looks like the numbers have sort of slowed down. I'm sure other people will join, but thanks for joining this webinar on this beautiful sunny day here in Vermont. I hope it's sunny where you are too. I'm Ali Kasiba. I'm Extension Assistant Professor of Forestry at the University of Vermont. And I just want to let folks know about the UVM Extension Forestry Program. It's new in the last year and a half. And what I do at the UVM Extension Forestry Program is to provide science-based information that really covers a gamut of forest health, forest ecology, forest management, including things like mycorrhizae and biodiversity and different aspects that we're all interested in. So I do have a lot of resources from partners and other folks and things that I've created on this website. And you can see that QR code, including a pamphlet that I worked with these two folks on, Amelia and Eva. It's a pamphlet on mycorrhizae fungi in the Northeast forest. So I will send a link to that to all registrants, but if you want to find that pamphlet ahead of time, go there. So a couple of housekeeping items before we get going. For foresters interested in continuing forester education credits through SAF, please add your name to the link in the chat. You can also check out that QR code. It's just a Microsoft form. You need your name and email address. Make sure it's the email address associated with your forester's license. So if you are a certified forester, that's important. I'll upload the list to SAF. I'll also be sending certificates to all folks that put their name on that list. And so the link should be in the chat as well. I'll put it in again after I'm done sharing, just to make sure. We also have captioning available. I did put the link in the chat. I'll put it in again for folks interested in live captioning. If you click on that link, it'll open another window with live captioning. For the Q&A session, we'll be using the Q&A box at the bottom, so please don't put questions in the chat. For our presenters, please use a Q&A box. And this webinar will be recorded. I'll be posting it to the UVM Extension Forestry YouTube channel. I'll send that link to all of you who are here and those who weren't able to make it probably in a few days. But so I'll send the pamphlet, that link, and to folks that need CFE credits, a certificate. Without further ado, I want to introduce our presenters today. So Dr. Amelia Fitch recently earned her PhD from Dartmouth College, where she studied mycorrhizal fungi in the Northeastern forest. She also holds a master's degree from the University of Cambridge and a bachelor's from the University of Oregon. She's currently working at the Oregon State University, examining how silviculture can help working forests mimic traits of old growth forests to buffer the effects of climate change. And we also have Eva Legge, is a first-year master's student at SUNY College of Environmental Science and Forestry. Her research focuses on how seedlings and mycorrhizal relationships are affected by different forest management techniques. She has a bachelor's in biology from Dartmouth College, where she studied mycorrhizal-mediated forest management. So without further ado, I'm going to pass it off to Amelia.
Great. Thanks so much, Ali. So I will do the shuffle where I now share my screen, get the PowerPoint going. Okie doke. Just need to move that out of the way. Cool. Great. Does that look okay?
Looks great.
Great. Okay.
Well, I should just say one last thing to folks.
Sure.
If you want to move the little window that shows the presenter video, you can move that around in the top, or you can minimize it if it's blocking any of the screen. All right, go ahead, Amelia.
Okay. All right. Sounds good. Okay. Thanks so much for joining, everyone. We're very excited to talk with you about mycorrhizal fungi and forest ecosystems. So to start, I will give you just a quick overview of what today will look like. I will give some background information just so we're all on the same page. And then I will go into the effects of mycorrhizal fungi on the below ground world, and specifically soil organic matter. And then we will do a share screen shuffle, and I will pass the baton to Eva, who will go into the above ground world of mycorrhizal fungi and the effects on forest communities. And then we will switch again and talk about the research component in Northeastern forests. Yoke. So mycorrhizal fungi are everywhere across all kinds of ecosystems, from the tropics to the Arctic. And below ground, what's actually happening, the mycorrhizal fungi, through these structures called hyphae, which are a lot like roots, but they're for fungi instead. They're connected to root systems, and these trees or other plants, they can obtain nutrients from the soil directly through their fine roots, or they can get nutrients from mycorrhizal fungi. And this is the primary way that the resource exchange is happening and why this relationship exists. And these mycorrhizal fungi, in return, are receiving carbon. They're receiving sugars and other molecules from these trees that have been fixed through photosynthesis, passed down through their roots to the hyphae, and that's where the mycorrhizal fungi get their energy. There are other fungi in the soil. There are pathogens. There are also this group called saprotrophs, and that just means that this is a group that gets their carbon from the soil organic matter. So they are decomposers. They're, the reason that we're not like up to our eyeballs in leaves and dead wood and stuff. Like they help break everything down and turn it back into smaller and smaller components that trees and other organisms can use, microbes. Okay, there are actually several different types of mycorrhizae, but the ones that we're going to focus on in this talk are the ectomycorrhizas. So you can think of like hemlock is a good example of a tree that associates with ectomycorrhizal fungi. And we're going to focus on the arbuscular mycorrhizal fungi, which are a type of endomycorrhiza. And I'll get into what that actually looks like and how these two are different. So ectomycorrhizae are kind of on the outside of the root. And so what that looks like, they weave these structures called Hartig nets, and they're primarily on the outside. And if you go out into a forest and dig up some soil, get some roots of a tree that associates with ectomycorrhizal fungi, you can actually see these woven structures because they're oftentimes different in their color and texture. They can look fuzzy, for example. Arbuscular mycorrhizae, again, they're a type of endo, so think like inside the root, and they actually form structures inside the root cells, and that is where nutrient transfer takes place. So some examples, again, I did have photos. Some examples of ectomycorrhizal trees that you are familiar with, like yellow birch, eastern hemlock, red oak, and for AM associating trees, we have sugar maple, white ash, and northern white cedar. Oops. Okay, a little bit of a lag. We're good. Oop, I'm gonna go back. Okay. I'm going to get into the below ground world a little bit now that we have some background information. So there's a lot going on in this diagram, but I'm going to start in the center with the mycorrhizal fungi. So we have mycorrhizal fungi zoomed out from a very tiny microscopic, what we couldn't really see with our eyes. And these fungi they're responsible as a kind of hub between all of these different processes happening. So trees are dropping litter to the forest floor. It's being incorporated as soil organic matter. And then mycorrhizal fungi are very important in getting the nutrients that are locked in that soil organic matter to the trees. And then the trees and the mycorrhizal fungi, they can affect this decomposition process of soil organic matter through their interactions with the other microbes living in the soil, and specifically the saprotrophs, those microbes that are decomposers of soil organic matter. And one of the primary ways that they do this is through this process called rhizodeposition. And here I have this word, it's exudates that are being transferred. So rhizodeposition, like rhizo meaning root, deposition is like, stuff raining down or whatever. It just means that like little bits of things from roots are ending up in the soil, and they're a really good substrate, really good food for these decomposer microbes. And these exudates in particular, they are sugars and other molecules that are super easy to decompose that are being transferred from the tree through the roots and through the hyphae of the mycorrhizal fungi out into the soil to those saprotrophs. And it's like giving away energy, like giving away food. But the bonus for the tree is that these saprotrophs then get this, very high energy, easy to decompose substrate, and they're like, wow, we can generate, we can help decompose the soil organic matter that's already in the soil and release nutrients, which is what the tree wants. It's all about getting those limiting nutrients from the soil for growth, reproduction, all of that. So I've broken down the main ways that these two main types of mycorrhizal fungi can affect soil organic matter. And I'm going to walk through each one of these, some in a little bit more detail, some in less. So first, starting with the litter quality. Often AM, so arbuscular, I'm going to start calling AM, and ECM, ectomycorrhizal, they tend to have different types of litter. And what I mean by that is that it's decomposability. So some litter decomposes really fast. Think of like ash litter, whereas some litter decomposes very slowly. You can think of like hemlock, and actually put some photos up here so we can see that there are these trends, and not every tree follows into this pattern, but there is a broad pattern where ECM associating trees tend to have slowly decomposing litter. Like think of the organic matter in the O horizon that builds up in a hemlock or a pine forest. Those are both ECM associating trees. Whereas AM associating trees tend to have faster decomposing litter. And that fast decomposing litter means that more nutrients are available in the soil. Whereas for the ECM associating trees or slowly decomposing litter, organic matter tends to build up and nutrients are often more limiting. And there are, this is a huge for the soil organic matter world in terms of like carbon storage from one perspective, but also nutrient availability and forest health on the other. So I'm going to get into something that's related, and I'm calling this the nitrogen. So I have it shortened to N, but it's nitrogen acquisition strategies. And what I mean by this is that, nitrogen is like it tends to be a limiting nutrient in these ecosystems. And it isn't for every type of forest. Like in the tropics, it's phosphorus. But here, like this is a really important kind of, theory and hypothesis about these different mycorrhizal types. So AM fungi, they tend to be scavengers. Their hyphae are often pretty short and compared to the ECM fungi. And the ECM fungi, they've actually retained the ability to actively participate in decomposition. So these ECM fungi, they're getting their carbon from the trees for sure. But like those decomposer microbes, they can produce these things called enzymes. And I've these yellow blobs here with the spiky bits. Those are my enzymes. And so what those are doing, they're taking larger molecules from the soil organic matter, breaking them down, releasing nutrients. And so these ECM fungi, they can forage greater distances, and they can actually seek out patches of organic matter and start to decompose them to get at those limiting nutrients. So for me, it helps to think about enzymes in terms of like my own digestion. So we have enzymes in our gut for breaking down our food, and these microbes, including these many species of ECM fungi, it's pretty similar. I mean, we don't have this exact same ones, but they're just secreting these enzymes out into the soil matrix, and then the decomposition happens outside of their tissues, outside their bodies, and then they like soak the nutrients and other things they need back into their cells. But everything takes place inside for us, which is really nice and handy. And a cool, enzyme example that we see is there's these enzymes in a lot of fruit like apples where they get in contact with air, and then this enzymatic process happens, and then we see a color change, which is actually a product of an enzyme taking a substrate and turning it into something different. Okay, so moving on to this, what I mentioned before, rhizodeposition, right? So rhizodeposition just means like stuff that comes from the roots. Could be little bits of roots, could be little bits of hyphae from the fungi, and it could be exudates ending up in the soil as substrates for the decomposers, the saprotrophs. And there's actually, there's a lot of debate about whether AM or ECM fungi and their associated trees have more or less rhizodeposition. I did a chapter in my PhD on this, and what we're what we're finding is that, or what we think is going on is that it really depends on like the nutrient and specifically nitrogen like profile in the soil. So if there's a lot of available nitrogen, the ECM and AM fungi and associated plants, like they behave differently compared to when nitrogen is really limiting. So I don't have like a straight answer for you, like, yes, one does more, one does less. This leads to more soil organic matter decomposition. It's just really complicated. So in this diagram, what I'm showing is that, again, we have this enzyme process going on in the lower right-hand corner where ECM fungi can produce their own enzymes, leading to more nutrient uptake potentially. They're doing some rhizodeposition, there are these sugars, I also called exudates. But these AM fungi, because they can't do this direct enzyme process, it might be an argument for the fact that they would produce, more sugars, more exudates to stimulate that decomposition. But it likely depends on what's already in the soil. Okay, and then lastly, I just want to spend a quick minute talking about necromass. And in this context, it just means like dead fungal tissue. So the actual tissue that these mycorrhizal fungi produce as they, grow, link up with more roots, it can be really important for soil organic matter stability, both carbon staying in the soil because of how difficult the necromass can be to break down. And it also can be really important for the other microbes in the soil as a substrate that they would, want to eat. So I just want to show you some photos from a student that we worked with in our lab. So these photos were taken by Zach Short, who is currently in Costa Rica on the Dartmouth Foreign Studies program. And he sent me, we're working on a paper together, and he sent me this figure that they made from a project where they characterized all the different types of ectomycorrhizal fungi that they found in, these particular soils. And they, don't have any internet where they are at this field station. So they were just they basically named them whatever they thought they looked like. I personally like, there's white and black Christmas tree. But yeah, so this just shows like the huge variety in growth forms that ectomycorrhizal fungi can have. In our muscular mycorrhizae too, we just can't see them with our eyes. But yeah, huge variety of growth forms and the potential for like really big differences in how the dead tissue then sticks around in the soil. So very cool stuff. All right. So now I'm gonna stop sharing and we're gonna swap over and I will pass it off to Eva.
Awesome. Okay. Can you all see my screen? Look good?
Yeah,
looks good.
Looks great.
Awesome. Great. Well, thank you so much, Amelia. It's been such a delight to work with Amelia for the past few years, and I'm really excited to tell you all about the effects of mycorrhizae on above ground plant responses, and then chat about some of the cool research in this realm that has been going on in northeastern forests, including some of our own research. So, one second. Okay. So over the past few decades, global change has really shifted the composition of northeastern forests. And these changes are projected to continue with increasing frequency in the years to come. And as I'm sure you all know, what makes it really tricky to address from a management perspective is that no one thing is really causing our forests to transform. And it's really a constellation of different stressors that have made the forests around us what they are today. Invasive pests are one of the biggest stressors shaping our forests. The hemlock woody algid, for example, is this aphid-like pest that literally sucks the nutrients out of hemlock trees. And the emerald ash borer has been decimating ash trees across the Northeast. The legacy of fire suppression ever since European colonialism has created this feedback loop called mesification that has created these monodominant maple beech forests as opposed to this complex mosaic of oak pine savannas and more mesic forests that were once more dominant across the East Coast and would likely be more resilient to future climates. Invasive plants, too, don't just lead to shifts in the understory, but can have ripple effects to the canopy. For example, garlic mustard is allelopathic, which means it can kill the symbiotic fungi like AM fungi, which Amelia was talking about, that associate with sugar maples. Pathogens, too, such as the fungi that cause beech bark disease, are leading to widespread declines in beech trees, and as well, the effects of acid rain can transform the chemistry of the soil, adding bucket loads of available nitrogen, but leaching out calcium, which is also essential for sugar maple growth. And then, of course, there's climate change. So repeated freeze-thaw cycles can damage root systems. Warmer and drier periods can increase a tree's drought stress, and earlier spring leaf out can cause really serious damage to trees. So you may be thinking, oh my gosh, Eva, there are so many threats looming to our forests. Why the heck do we see so much green? Well, at least in the summer, not right now. The answer is, first of all, that trees have an incredible array of adaptive mechanisms to deal with all these stressors. But that isn't the only reason, because as we know, the forest isn't just what we see above ground, but in fact, there's this entire world below ground that really modulate forest ecosystem response to global change. And that's a picture of me out in Corinth, one of our research sites, pointing to the vast soil profile beneath the surface. And so the most important symbiosis for our purposes, as we've been discussing, is mycorrhizae. So as Amelia's briefly mentioned, mycorrhizae can act to ameliorate a tree's drought stress. It can transfer limiting nutrients to trees, and can also protect the trees against pathogens by actually sheathing the root tip and colonizing them preemptively against potential fungal pathogens. And we've already seen these symbioses shift due to climate change. So over the past few decades, there's been a major shift in the dominant mycorrhizal associations in the Eastern US towards AM associating trees. So again, that's sugar maple, ash, and cherry, among others. So this figure is this really cool study that was published in 2019 in Science that represents the shifts in AM tree dominance over the past 3 decades using a long-term FIA data set, and accounts for more than 3 million trees. So the red indicates a shift towards AM associating trees, whereas the blue indicates a shift towards ECM associating trees. So overall, there has been a shift in the past few decades towards AM associating trees. In addition, they surveyed sapling to overstory tree ratios, and they found that there is projected to be an even increased shift towards more, especially maple trees in the future, and a widespread decline in oaks, which is very worrisome. And so there's 3 main drivers of this that they mentioned in this paper: nitrogen deposition. AM trees are competitively superior to acquiring inorganic nitrogen, like Amelia mentioned. Fire suppression, which has led to forest mesification, as I mentioned, as well as climate change, so increases in mean annual precipitation, for example, is often associated with increases in AM tree dominance. However, if you actually zoom into the Northeast, these patterns are actually reversed. So you can see there's a lot more blue here than red. And that's really driven by the fact that there aren't that many AM associating trees in the Northeast. So any change in one or a few tree species, or even in a certain genera of trees, can really have a huge effect on this shift in mycorrhizal associations. So as I've said, the main AM associating trees in this area are maple, ash, and cherry, especially in New England. And ash trees are being decimated by the emerald ash borer, and sugar maple has been under threat from acid rain, calcium depletion, as well as just the fact that climate change is not going to particularly favor this species. So it's a hard balance. They are very dominant, despite the fact that they won't be projected to be successful in the future. And in the Northeast, you can really see that this change in just a few species is really driving changes in AM to ECM ratios. Okay. So it's really important to understand how below ground networks are affecting specifically seedlings' response to these different stressors. And despite what is often touted in the media, seedlings' access to these below ground root hyphal networks doesn't just have positive effects on seedling success. So there are positives, as I've mentioned, such as nutrient provision for mycorrhizae, water provision, and potential for resource exchange with neighboring trees. But there's also negatives, exposure to pathogens from conspecifics or neighboring tree species. So AM fungi especially have a low capacity to protect seedlings against pathogens since they don't completely sheath the root like EM fungi do. Mycorrhizal parasitism. So this is especially important and has been discussed recently in various papers debunking the idea that mycorrhizae are just mutualistic. So in high light or high nutrient, low light or high nutrient conditions especially, they can become parasitic. And competition for water and nutrients, so between roots and between actual mycorrhizae, can lead to declines in a seedling success. So all of these different network effects, as I call them, really act simultaneously to shape seedling success and, in general, the success of any tree's growth and survival and acquisition of nutrients. But that doesn't mean that the balance of net positives and negatives are always equal. So each force's relative contribution is really likely to change based on a number of different factors. And one of the things we want to discuss is how management can affect these various below ground contributors to above ground tree success. So now we're going to dive into some of the research that's been done in the past on the effect of management on mycorrhizal symbioses in Northeastern forests. So here we have some really fascinating research that occurred in the University of Vermont's Adaptive Silviculture for Climate Change, or ASK sites in northern New Hampshire, on the effects of management on various fungal types. So they looked at ECM fungi, parasitic, and saprotrophic fungi. We're going to focus on the ECM fungi. So what they did was they performed spore carp, so mushroom surveys, to assess fungal species richness, which is the number of different genera, as well as the diversity and biomass of these fungi in 0.4 hectare patch cuts, as well as intact forests. So forest that hasn't received any specific cutting, at least in the past few decades. So there's a lot going on here, but we're focusing on ECM fungi here. And so you can see that 18 months post-harvest, ECM fungal richness and biomass was significantly reduced. And that gives us some insight into potential recovery times for ECM fungi following harvest, and future studies that look maybe 24 months post-harvest or 36 months. It would be really interesting to see how these ratios might shift. So this next figure shows some really cool research that also occurred up in the ASK site, so in northern New Hampshire. And it was led by Dr. Ryan Stevens, and he surveyed rodent scat. And he found that rodents are, in fact, incredibly important vectors facilitating AM and ECM spore dispersal in patch cuts. So this capital A through G, this top row here, are AM fungi, and then all of these are different ECM spores. And they found that while AM spore dispersal was really redundant. So there was lots of different vectors for AM fungal, spore dispersal, ECM dispersal networks really relied heavily on this one guy right here, Mycodes gapperi, which implies that management aimed at maintaining ECM fungi following a harvest might also want to work to maintain specific species of rodents. And then finally, we have this schematic showing a potential direct application of mycorrhizal research and the direct application of mycorrhizal fungi into Northeastern forest management. So this really looked at the role of ECM fungi in temperate forest restoration, but it could be applied to AM fungi as well, I think. So they discuss how soil transfer from mature adjacent forests could provide potential inoculum for seedlings. And another paper with the same author, so Horton's on both of these papers, found that ECM seedlings, which were inoculated with soil from old growth ECM forests, really did better than those that weren't inoculated. And then they also proposed potential benefit of planting seedlings near remnant trees that might provide potential inoculum sources as well, which we will also discuss. But I think one of the main takeaways from this paper is that the number of studies in this realm is incredibly lacking, and there's a great opportunity for future research in this area, which I'm excited for us to discuss as well for our research. So building off of these knowledge gaps that we've established, we had a couple of questions when going into our own research experiments. First, how does seedling survival and growth differ between forests previously dominated by AM and ECM associated trees? And second, how do species of seedlings differ in their growth and survival with or without access to mycorrhizal networks? So with that, I'll pass it off to Amelia.
Cool. Thanks, Eva. That was awesome. Okay, it's loading. Great. You should be able to see this slide now. I will be focusing on the first question about how seedlings survive and grow in logged gaps where the trees used to be AM or ECM associated. And so this project couldn't have done it without being supported by Dartmouth, of course, and the University of Vermont, specifically Tony D'Amato, and the community in Corinth, and also funding from the Northern States Research Cooperative. So very, very, very thankful for that. So in this project, I'm just going to kind of center us on some framework first. We know that there are lots of disturbance mechanisms from what Eva was just talking about: climate change, invasive pests. Timber harvesting is something that we use to get wood. I mean, wood is a very great resource and we need it. And so understanding how forests under the threat of these other kinds of disturbances are going to interact with timber harvesting is something that's super important. So these disturbances have the potential to create changes in below ground microbial communities, which could have implications for how seedlings are going to have access to mycorrhizal fungi, potential risks of pathogens in the soil, and these can create differences in seedling survival, potentially depending on their mycorrhizal association, which is what this project gets into. And this has implications for how forests are going to regenerate going forward. So in this project, we had 16 total plots, and 8 of them were our control plots and 8 of them were quarter-acre logged gaps. And so we went out into the forest to set up these plots, and we found areas, quarter-acre areas, where we had a dominance of either AM-associated or ECM-associated trees. So on the AM-associated side, in Corinth at least, it was very much ash, and maple. And then on the ECM associated side of things, it was more of a mix, but tended to be hemlock, birch, and beech. So anyway, so in these logged gaps, so for each mycorrhizal, I'm going to call this a legacy now. So when the forest plots were harvested after timber harvesting, there is a kind of legacy left behind, and we wondered how that legacy, depending on the mycorrhizal association, would affect the seedlings. So we chose 8 species of seedlings for each mycorrhizal association and planted them in these locked gaps. And I'll get into some more details. And this is a bird's eye view of what the Corinth experimental forest looks like. There's a much bigger experiment going on here with Tony looking at silviculture practices in emerald ash borer. So there's many plots in each of these units, but we only used a smattering of them. So these control units are green and the harvested units are orange. And then the AM and ECM dominance is indicated by a circle or a star shape. We have 16 total plots, half each AM and ECM associated, and then half of each of those were either legacy because they were harvested or they were control. This is an example of one of our AM legacy plots. You can see the seedlings are all in cages. This is actually one of my favorite plots. It's really pretty at a certain time of day. Well, any time of day, but I think this was taken during golden hour. And then, so the planted tree species, for the AM associated side of things, we had red maple, sugar maple, black gum, and black cherry. And then for the ECM associated seedlings, we chose bitternut hickory, black birch, American basswood, and red oak. So to start, I'm going to talk about the fungal biomass itself and then get into the effects of the legacy on seedlings. So for these data, we were looking at both the control and the legacy, or I guess I've labeled it as logged plots. And so on the y-axis, we're just looking at AM, arbuscular mycorrhizal fungal biomass. And then the different colors in the plots represent the legacy type. So AM fungal biomass is being represented here in both the orange, which is the AM legacy, and the blue, which is the ECM legacy. So we're comparing AM fungal biomass in all of the 16 plots. Cool. So I found this can be confusing. So there's a lot of different, like AM this, ECM that. So what we found comparing the control to the legacy plots after harvesting, so we had, sorry, just to back up real quick, we took soil samples before the treatment plots were logged. So before they became a legacy, we took soil samples and we were able to measure the AM fungal biomass to compare to what happened after the harvesting. And so after harvesting, we saw that there was a 79% decrease in AM fungal biomass. And so this, as you can see, this is just from, 2020 was before, and then just one season, just 2021. So this was like, fall of 2020, I think in October, and then this was taken in June, and then again in October. And so actually just in the past couple weeks, maybe even less, I finally got the data back from the lab that runs the samples for me. And so I have the 2022 data, and I'm sorry I didn't have time to get that up here for you, but I'm happy to send out an update through Amelia or whatever. We can figure that out at some point. But I do have data coming in, and I'm very curious to see how a year looks for the recovery of the AM and ECM fungi. So now let's look at the same thing, but for the ECM fungal biomass. So again, we're the y-axis, ECM fungal biomass. We're looking at control compared to the treatment, the legacy plots, and the legacy plot type between AM and ECM. So we also found a decrease post-harvest for ECM fungal biomass, but it was much less drastic. It was only a 35% decrease in biomass. And I have some theories as to why, that if I have time, I will get into a little bit. So just save, bookmark this for now. Just want to make sure we're not going to run over time. Okay, maybe. So now let's take a look at the seedlings. So seedling survival on the y-axis here between, and now we have this plot is split by legacy type. So we have AM legacy on the left and ECM legacy on the right. And on the x-axis, in each of these legacy types, we have AM seedlings planted and ECM seedlings planted. And so AM seedlings, when they were planted in AM legacy plots, they had what I'm calling a home field advantage. So they had this slight increase in survival, but the ECM seedlings as a whole, it did not matter where we planted them. Oh, and I should have mentioned just real quick that this was including 2 years of data. So like a full year had passed at least. And one of the reasons why we think there may be this AM home field advantage is the way that these two different mycorrhizal fungal types, interact with the nutrients in the soil. So kind of getting back to those nitrogen acquisition strategies that I was talking about, AM fungi tend to do better, they tend to be better at getting nutrients when they're already in an available form. So what we found here and what I'm showing, this is actually growth, it's just height on the y-axis. And then on the x-axis, we have soil nutrient availability, and this includes 2 different kinds of available nitrogen. And for AM seedlings in this red line on the left, there was an increase in growth when they were planted in AM legacy plots, but not in the ECM legacy plots, and it did not matter for the ECM associated seedlings. And so there might be something to do with nutrients here. Do I have another slide? Okay, yes, I do. So what might be happening, and we have some more like a bonus study in the works to get at more of this mechanism of like, is it the nitrogen? Is it the AM fungi getting the nitrogen? What we think might be happening is that shifts in below ground communities, there could be differences in access to mycorrhizal fungi, or potentially just differences in how these kind of early colonizing mycorrhizal fungi to the roots could be behaving and interacting with the seedlings that were planted. And so this might be why we're seeing differences in seedling survival and growth. And what we're doing now to get at that is measuring the nitrogen in leaves and in the soil of like each individual seedling, and then they're called stable isotopes, and I don't have time to go into them, and I wish I could because they're so cool. But basically, like we might be able to partition the mechanism of like, is that nitrogen potentially coming from a mycorrhizal source as opposed to just from the soil. All right. So now I'm going to pass it off to, Eva, to go into your research project.
Great. Thank you, Amelia. So going back to this framework that Amelia has laid out beautifully, I really like this graph. My research aimed to look a little more closely at how seedling access to mycorrhizal fungi and surrounding tree roots may change with management and result in seedling success. So for my study, I looked at 3 different AM associating seedlings, so sugar maple, black cherry, and black gum, all of which may be managed for in various climate adaptive silviculture techniques in the Northeast, but for different reasons. So sugar maple is currently the most abundant in this region. It's shade tolerant, adapted to our current climate, but expected to decline in future scenarios. But it really holds incredible cultural value as well as timber value. So it's an important species to maintain in our forests. Black cherry, on the other hand, is a more southern adapted species. So you can see that my study site is kind of at the northern edge of its range. So any effort to increase the population in this area really consists of a population enrichment treatment. So increasing the population at the northern edge of its range to prepare for future climates. Whereas black gum is an opportunistic shade tolerant species that has been proposed as a new species. for whom New England would be a suitable habitat by around 2100. So this species is more of a candidate for assisted migration in my study area. And you can see it's just kind of out of range. There might be some, but they would be more swamp dwelling species. So my research occurred in the same area as Amelia. And this was for my undergraduate thesis. And so I was highly inspired and influenced by Amelia's work, and was done after I came out in a field season with her in 2021. And I was astounded by the work she'd done. So I wanted to do this little experiment building off of it, looking at planting seedlings in patch cuts, so those quarter-acre patches, as well as selective cut plots. So this is single tree selection, where they cut just ash trees. Remember, this is part of a big adaptive silviculture for emerald ash borer experiment. So that's kind of the precedent for this management type. And all these areas were previously dominated by AM associating trees. So I was just looking at these AM legacy sites. So I planted clusters of 12 cherry, sugar maple, and black gum seedlings. So 4 of each seedling type in each cluster, at a range of access to below ground networks. And all of the plots were about 0.75 meter radius. So about large hula hoop size, really, really large one. And the 3 different experimental treatments were trenching. So this represented the lowest amount of access to the roots and hyphae present and pathogens present on neighboring trees. And we did this by trenching down to a depth of 45 centimeters or until we hit bedrock, and then lining it with a thick polyethylene sheeting and backfilling it. And this is a pretty early type of technique used to isolate seedlings from networks. So it's been done in various methods for the past century or so, specifically looking at mycorrhizae. And it's definitely labor intensive, but it definitely has an effect. And then we did a root severing technique, which represented a medium amount of network access for seedlings, in which we pierce the soil in a circle around the seedlings to the same depth and also the same radius of the plot. And that represents a temporary severing of the roots, assuming that they'll regrow, but we did reinforce it throughout the summer of 2022. And then the control plots were dug to a depth of 45 centimeters and reinforced. The control plots were not disturbed at all. So the measurements I took soil samples, dug root pits to assess the density of live roots in the area. I measured the growth, survival, and foliar nutrients on my seedlings, as well as took some destructive harvests of sugar maple in the control plots to compare mycorrhizal colonization between the different management types. So first of all, we found that live root mass was higher in selective cut plots, which is kind of just confirming our assumption that there was a higher density of roots in the area where there's less disturbance. And so one of the cool results we found was that when we dug up the maple seedlings and assessed the colonization of AM fungi on the roots, we found that the maple seedlings planted in the selective cut plots had significantly higher AM fungal colonization than in the patch cuts. So you can see it's about 50% colonization versus 25% colonization in the patch cuts. And then the colonization of AM fungi plus this kind of fungi called dark septate endophyte. So this isn't technically mycorrhizal fungi, but it's been shown to potentially have positive effects on seedlings. It was also significantly higher when those 2 fungi were pooled than in patch cuts. Corresponding with this higher colonization was a higher survival of maples in the selective cuts. So about 90% survival versus 2/3 survival in the patch cuts, whereas the black gum and the cherry had no differences. And then when we assess the growth of the seedlings, you can see that in the patch cuts, cherries had significantly higher growth, as well as significantly higher growth with increased exclusion from networks. So this is the trench plots, these are the controls, whereas there was little change in the cherries and the black gum. And then for foliar nitrogen, which is a proxy for plant vigor, they're able to acquire more nitrogen, can assume that they have higher functioning capacity for future growth and survival. Cherries had higher foliar nitrogen as well, with increasing exclusion from networks. So you can see that their growth patterns look very similar to their foliar nitrogen patterns, which is very interesting. And then the maple nitrogen was also higher in the selective plots, where they also had higher survival and higher mycorrhizal colonization. So this initial window into their performance and colonization gives you a window into how each species might cooperate with below ground networks in different management types. So sugar maple demonstrated higher survival, mycorrhizal colonization, and foliar nitrogen in selective cuts, while black gum really did not seem to have changes in their growth or survival with these changes in network access. Whereas black cherry really benefited from having exclusion from below ground networks, in addition to being planted in the patch cuts. However, I did want to mention that this is data from one growing season. And as Amelia mentioned, things can definitely change when you get in new data, you start interpreting it, and you realize that, of course, things shift. And so looking at in this past year of growth and survival really looks like the potential negative effects of patch cutting on maples in the first year kind of balances out by the second year, which might point to the fact that the networks and the seedlings really do recover quite rapidly, because at the end of the day, a quarter-acre patch isn't that big. And so I did want to note that I'm really interested to see how these patterns continue to change over time. And this is just a very initial look into how management can change these mycorrhizal relationships. But future work is really necessary, and you will all just stay tuned for both of our work when it gets published eventually. So with that, I just wanted to say that these results really highlight how mycorrhizal fungi act as a bridge between the effects of forest communities on soil and the effects of soil on resultant forest communities. And as these studies have shown, if nothing else, it's incredibly complex. But the one thing we know for sure is that mycorrhizal fungi have a vital role right there in the middle and really mediate these reciprocal effects of forest communities on soil, with vast implications for management that we're just beginning to get into. So with that, we wanted to give a huge shout out to all of our collaborators, Tony D'Amato, Caitlin Hicks-Priest at Dartmouth, Allie, who's been amazing, Kevin Evans, who's the Dartmouth forester, and none of this management in Corinth would have happened without him. Other folks at UVM Extension, as well as the amazing field crew that helped Amelia plant all of her seedlings. As well as we wanted to thank the other amazing members of our lab, especially Sarah, the lab manager, and all the great undergrads and the amazing Waldo the dog as well. So with that, we would love to welcome any questions. And we also wanted to leave you with a question of our own, which is, how would mycorrhizal fungal research be useful for a management issue you are facing? And please feel free to post it in the chat or email us, or both. We would really love to know.
Wonderful. Thanks so much. That was great, Eva and Amelia. I guess I'd say stop sharing your screen so we can see you both, and I will ask some questions. We're getting a lot of comments about just fantastic presentation, lots of information in there. So as a reminder, this is recorded. If you want to watch it again, maybe on like halftime to absorb all the information, take notes. This was a lot of really great data and research. So I'm going to kind of go through some of the questions that are asked here. I did schedule this for to go till 2:15. So if folks can stick around, and I knew that there would be lots of questions and a lot of information. So first, sort of like a general question that came in early, can you talk about host specificity with mycorrhizae? So, you know, is one species of mycorrhizal fungi always associated with a certain species of tree, or can they be associated with multiple species of trees?
Great question. Eva, do you want to, I feel like I'm happy to answer it, but I feel like, yeah, your research is like way more in this direction.
Oh, sure. Yeah, so that is a really great question. And we're honestly, I feel like, just hitting the surface. By we, I just mean mycorrhizal ecologists in general. There's still a lot of open questions in that regard, but we have begun to build these frameworks in figuring out how host specificity changes among different mycorrhizal types. And so, short answer, absolutely exists. And I would say it's a little more prevalent in ECM fungi. So pines are a great example of host specificity. They are incredibly reliant on, and all of the Pinaceae really reliant on ECM fungi for their survival. And they have quite a unique host of mycorrhizae that associate with them. So the colonization on a pine tree versus an oak tree that's growing right next to it is probably vastly different, which is something I'm interested in looking at in some of my master's research. So AM fungi, there's definitely host specificity, probably less so. They're known to be a little more generalist, but that's being called into question too, so a little complicated.
I also, oh, sorry. Just to rock everyone's world a little bit, during my like dissertation, like the closed door portion, so I do the presentation for my dissertation, and then I do the closed door portion, just my committee. During that closed door portion, one of my members brought up to me that there are people in Europe, especially, who do mycorrhizal research, calling into question whether or not they're actual mycorrhizal types, which is like the premise of all of our experiments and this framework. So they're like, some people have found like what we think of as an AM fungal species like in an ECM associating tree too. So like there is still so much that we don't know. And yeah, with ECM fungi, I think there was a specific question about, I think it was I can't find it, but yeah, there are some species that are specific, and then there are some that are more generalist, and we don't know why, but it's very cool.
Always good to know that there's lots to still research on this topic, which is true for most things in forests. So the great question came in about sort of longer term impacts in our forest, right? You all are talking about sort of shorter term impacts. What about the impact of our land clearing that happened here in the 1800s and really even into the early 1900s? Sometimes, you know, there were grazing animals, there was a disturbance of the soil and plowing. How do you think that affected mycorrhizae and sort of the recovery of our forests?
That's a very good question. I guess I don't know, any studies that have looked at that. I don't know if it would even be possible to be able, we wouldn't have soil samples from way back then. I guess I think these forests are pretty resilient. I don't know. I mean, the work that Eva talked about from like Ryan Stevens and those folks looking at how in clear cuts rodents are really important for bringing in spores. Like hyphae are like really amazing at like getting into soil and like helping. I actually didn't go into this, but they're super important for helping to create soil structure. So I feel like it probably wouldn't take that long. I mean, that long, maybe, I don't know, 20 years, 30 years for soil, like the hyphal structure of soil to start to resemble what it did before, but it also probably depends on what's coming from the trees above too. So I don't know, I'm just like pontificating now, but it's a great question, like deserves more attention. Yeah, what do you think, Eva? Do you have any thoughts?
Yeah, it's a really great question. And I have a couple thoughts on it. First of all, I think the major land clearing, there was clearing for agriculture, and this in the 1800s kind of slash and burn. So there were some more fires in the forest after all this kindling was created. But after that, starting maybe mid to late 1800s, this vast effort of fire suppression. So fires were really dominant before European colonization by indigenous communities and completely eviscerated after colonization due to, well, I could go more into this, but it's really created when the forest did recover. So the land was abandoned, people moved west, like farming here. Why would we farm here when we could go out to the Great Plains? The forests that did recover were not fire adapted forests, and they were these monodominant, as I've said, maple beech forests. So it kind of was a bit of a reset, and I think was a major driver of mesification and the increased dominance of this AM associating forest versus the ECM associated forests, like these oak pine savannas that used to go across the East Coast. And then in addition, I just wanted to note that most 80% of vascular plants are AM associated. And so we still don't know a lot, as Amelia said, about the actual effects of land clearing, how quickly it would take an intact older growth forest, whatever that means, to get back to that same mycorrhizal community. But AM fungi, as we said, are generalists and associate with those crops that would be farmed. And so in that sense, I think it would make sense that that would facilitate also AM tree succession as well.
Great. Okay, so I'm kind of lumping a couple questions together about stressors in the forest. So specifically, I was wondering, I know that at the Corinth site, there are earthworms and there are non-native invasive plants. And earthworms can sort of tunnel through soil, right, and disrupt that soil structure. And then invasive plants have roots that go into the soil. So how is there research, or do you, either of you, have knowledge about how those two stressors could impact mycorrhizae and sort of the processes that happen in the soil?
Yes, I can start out by talking a little bit about earthworms. I answered some like methodsy questions just like in the chat, and there was one question about what soil I sampled for the AM and ECM fungal biomass portion of the study. And I mentioned worms, and that we sampled an OA horizon because in many places, especially the maple and ash dominated sections of the forest, there was no distinction. It was OA and then it was B. It was like this kind of, gray, but also organic, not gray, but like, yeah, it was all mixed together. And when I first went out and sampled with the corers, I I was like, oh my, I'm like looking for the distinct soil horizons. And I was like, this is really confusing. Like these worms are messing up my ideal way that this was going to go. Yeah, so worms, I don't know a lot about them, but I just from like helping out with some teaching labs at Dartmouth, we do know that they increase soil respiration because they're bringing a lot of organic matter in from like the litter layer and bringing it down into the soil profile, which if you're interested in growing food, like that's a really good thing. Like worms are great for farms, but in a forest, it like they can be thought of as like ecosystem engineers. I mean, they are seriously changing the soil. And one way, and this is, I'm like totally kind of going off on a tangent, but when the litter layer is pulled down by worms and that really disappears, there's less room. There are fewer places for arthropods to hide, like so different insects, and so that actually scales all the way up to like the bird communities have, fewer things. The birds that are relying on these litter dwelling arthropods are like they're affected too. So there's this like cascade of things that worms can affect. And also like definitely in the soil, like increasing soil respiration. And I don't know what the worms would do. I mean, I can imagine that the worms are definitely, they are changing the structure of the soil and, munching through all the different layers of hyphae. And so I imagine it's just like this battle of like the worms come through and then like the hyphae grow back and then like the worms come through again. So probably makes it even more variable in the soil than what it would be otherwise.
Yeah, I think Amelia did a great job. I am no worm expert. I did just think there are these cool things called nematode lassoing fungi. We should really come up with some invasive worm lassoing fungi to deal with the invasive pest problem. So someone should get on that.
Any thoughts about non-native plant, like herbaceous plant understories that are non-native, barberry, honeysuckle? Has anybody looked into the impacts of those?
When it comes to mycorrhizal fungi, I don't actually know. I don't think so. I mean, especially in the Northeast, like we're really just kind of getting started with mycorrhizal research there. Yeah, I don't know. Sorry, I don't really have a better answer than that. I would be curious though, because in our plots, we have this one plot that we call the Mullen plot because it was the first couple of years, it was just chock full of mullen and it was the worst thing ever to do field work there. And like that, it was so different from the other plots. I was like, there's gotta be something happening to the soil here that maybe isn't happening in other places. Maybe it just had to do with like a dispersal mechanism. But I think it would be super cool to look at invasives and how maybe there are a type of fungi in the soil that are facilitating that invasion, or maybe those plants are so good at growing anywhere that they can just pop up, like garlic mustard. We've started noticing an influx of garlic mustard into the Corinth area too. I think I don't know where it came from, but anyway.
Yeah, and then there's the can of worms of that you could actually have non-native mycorrhizal fungi at sites. There's a researcher looking at that in South America that they've been imported in seedling containers with the soil for reforestation efforts. So they inadvertently so that adds another level. Okay, so thinking forward, particularly in Vermont, we've had a large increase in precipitation with climate change, and we're getting these really big punctuated rain events. What are your thoughts on, fungi love water, right? We saw a lot of fruiting bodies of mushrooms this summer. A lot of people noted that. What do you think for climate change will affect mycorrhizal fungi in the area? Do you think there will be an impact, positive, negative, varied?
Yeah, I can start and just as I've mentioned about the whole feedback loop of mesophication. So there's fungal effects on above ground plant response, but then there's also above ground plant effects on below ground, and it's just this loop over and over that's constantly reinforcing these transitions, as I've said, mostly towards AM dominance. And for the most part, AM associating tree species are a lot more moisture loving species like maples versus, let's say, oaks and pines. And I do think that there are various mechanisms that below ground fungi might benefit from it as well, but I do think it might start above ground, then it changes the below ground community and continues to.
Yeah, I think that it's very likely that some species will be better prepared to deal with the changes. And there are some studies. There's one person who's still in our lab getting her PhD right now, and she's doing a study on how how rain events are affecting like the microbial community in general, and how they process carbon and respire, and how much carbon's left in the soil. And it's a very like kind of like geochemistry focused perspective. But I think I was just thinking about like there are places that are doing rain exclusion experiments, and the ability to like add more rain. So I think it would be so cool to look to, and maybe someone's already doing this, I have no idea, but there are experiments out there where you can look at what's going to happen with more precipitation or less precipitation, and the fungi in the soil. Our former advisor, Caitlin Hicks-Prees, is doing a warming experiment at the skiway to look at the effects of freeze-thaw cycles on carbon in the soil. And I think similarly, it would be really interesting to see how the fungi change. And I don't know if she has plans to do that, but I can ask her when we meet next week.
I know she worked it into the grant, and then they said,
oh, cool.
There's too
of course
much you're looking at. So
yeah.
axe it.
Yeah, if there are any future graduate students on this webinar, there seems to be a lot of research topics on this in this area. So looking at the time, I think we'll call it. We do have a lot more questions, and Amelia and Eva can look at these, and if maybe we'll send some responses if they feel like there's ones they want to respond to. But thanks everyone for attending. Again, I'll follow up, probably not today, but on a day or two, with everyone. Link to the recording, the pamphlet that we all worked on with Tony D'Amato and Caitlin Hicks-Prees and some other folks, and some other details. So again, thanks, great questions. Thanks for the interest and sharing this. And thanks to our presenters, Amelia and Eva. Great work, very interesting, fodder, lots of fodder for future questions and research. So thanks for sharing your research and time.
Yeah, you're welcome. And again, there will be more results coming in, so we will figure out a way to send some updates like about the fungal biomass thing in particular.
Yes, fabulous. All right, take care, everybody.
Thank you all.